Welcome. My name is Stefan Howorka. I'm a professor in chemical biology at the Department of Chemistry at and I'm very glad, and I'd like to thank Conrad from Nanion for giving me the opportunity to talk here. Okay, the title of my talk is Bioanalysis and DNA-Sequencing with Nanopores, and I would like to show you some of the work which was done in my laboratory, and I would like to present in general also the sensing approach and why it might be something useful and good. Okay, let's take a step back. The sensing of biomolecules is generally relevant. One can think of the diagnosis of patients, for instance here with the microarray, and the diagnostics will help to find personalized medicine, such as, for instance, to tailor drug treatment, cancer treatment to the specific person who's got a specific type of cancer. Other reasons why sensing of biomolecules is relevant is environmental screening, but also to maintain high food standards. There are a couple of molecules type which should be detected. So on the left is a representative of the small molecule organic chemical class, in the middle is DNA, and on the right is protein and antibody. And I will mostly talk about the middle and the right one, so the DNA sensing and sequencing and the protein sensing. Existing biosensing approaches can use, for instance, HPLC or mass spectrometry. These are extremely good kits, highly reliable technology, but as you can see, they're sensitive, specific, and compatible with many analytes. However, one of the drawbacks of these biosensing approaches is that they're not really that inexpensive. They're not small and handheld, and often they are not easy to use, requiring a specialized person to carry out the analytical procedures. So there is a demand for new biosensing approaches, and these should be not only specific and sensitive, but also compatible with many analytes, inexpensive, small and handheld, and easy to use, and in the best case, they are compatible with personalized, portable computing equipment like smartphones. The question is, can this nanopore sensing approach meet the demand? The nanopore sensing approach is illustrated here. You've got here a slab of a membrane. It happens to be in this in this rendering a glass slide, and you see that the DNA molecule is being threaded through. In the middle, you see the same thing, basically now from the side, and this little hole here represents the hole in here. So this membrane has a hole, and if we zoom in, we see basically that ions are passing through, because we've got this membrane is separating two chambers which are filled with electrolytes such as potassium or chloride. And if a potential is being set up, ions are moving through, and the current can be recorded as a function of time. If analyte molecules pass through this opening, then the current blockade is current blockades are being generated. You can see here that a small analyte molecule leads to a small blockade, a larger analyte molecule leads to a larger blockade. It is also possible to engineer artificial binding sites inside this pore, inside the opening that's represented by this banana, and this black banana-shaped structure is a receptor which is able to bind complementary cognate analyte molecule. So that's a nanopore sensing. This approach here, right, is also called stochastic sensing. What are the nanopores composed of? The nanopores can be composed of inorganic materials such as graphene, molybdenum disulfide, silicon nitride, polymer. This is a side view. Here's a polymer. The small things in here, this is water. Or it can be a glass nanopipette. Another class of nanopores are membrane nanopores. The generic structure is illustrated on the left-hand side. It's a barrel in purple, and this barrel is inserted inside a gray membrane. Can be a lipid bilayer membrane, a biological semifluid membrane, but also a semifluid membrane composed of polymers. In order for this barrel to insert inside the hydrophobic membrane core, the barrel contains hydrophobic belt in orange. There are many classes of membrane nanopores, such as proteins, peptides, but more recently also structures composed of folded DNA, but also small organic molecules can be built up in supramolecular chemistry to form a barrel. The nanopore sensing was pioneered with the protein pore alpha-hemolysin that's shown here. You see from the top that the protein pore is composed of 7 subunits, and the side view shows that here a bottom stem, this beta barrel is inserted inside the lipid bilayer. The top part, the cap, is not inside this bilayer. The protein pore is produced by a bacterium, and the job is actually to kill host cells and to lyse the erythrocytes, hence hemolysin. But it was repurposed and retooled for nanopore sensing. So far I've given you an introduction about the need for sensing techniques which are able to accomplish sensing with small portable devices. I've introduced also nanopore sensing, introduced one specific pore, the alpha-hemolysin, and some also others. And now let's ask, is it possible to use the DNA? Is it possible to detect DNA? So DNA is the carrier of genetic information. We see here a beautiful structure, the double helix. The stuff here in the backbone, that's the phosphodiester backbone here, and here's the chemical structure. Phosphates here, ribose, and the DNA strand is the DNA duplex is composed of 2 strands, and these 2 strands are held together by Watson-Crick base pairs, and that's here is a mononucleotide of a G base. Okay, can the protein pore alpha-hemolysin be used to detect DNA strands? One approach to sense DNA with this stochastic sensing principle is illustrated here. You see on the left the protein pore, that's a cross-sectional view. The bottom part of the pore is inserted inside the lipid bilayer membrane. The top part here has been engineered in order to sense DNA. You can see here an engineered residue, a cysteine, which carries a DNA oligonucleotide. And the DNA oligonucleotide is schematically also represented here as a red strand. When this single nanopore is inserted into the membrane, one is observing this current. If a complementary DNA oligonucleotide is being added, this green line here, then these current blockades occur, indicated by B, and this represents the state when the green oligonucleotide has bound via Watson-Crick base pairing to the red strand. And this binding is temporary, so the DNA binds here, comes off, and so on. Now that's good, but the drawback is that in the previous case, the knowledge of the DNA strand was required. However, it's much better to read DNA without prior knowledge of the DNA strand. And the question is it possible to sense nucleotides? The receptor molecule, in this case, is a cyclodextrin. It's a ring-shaped carbohydrate which sits and can be placed inside this protein pore, alpha-hemolysin. And the purpose of putting this cyclodextrin adapter inside is to sense and detect the binding of nucleotides, the monophosphate nucleotides. And here is a classical readout trace, or what happens when a mixture of those four nucleotides, G, T, A, and C, are being added to this solution, and the DNA nucleotides, they go inside this opening and temporarily bind to the receptor inside this protein pore. And the readout trace is shown here, current versus time, and you can see different short current blockades which reach to different levels. For instance, here is a red line, and then there is a blue, green, and gray, and that's all color-coded. So each of these blockades represents the binding of an individual nucleotide inside this part here of the nanopore, and then they block the current to a characteristic extent. So if thousands of these events are recorded and then plotted in a histogram, then you get something like that. You've got then a very good discrimination between, let's say, G and C, the two of the four base letters. But T and A discrimination is not very good, as you can see here. Nevertheless, that experiment has shown that it's perfect, it's possible to read individual nucleotides. Now the next question is it possible to read the DNA or read the bases inside the DNA strand? The issue is when one is using DNA strands, that the strands are moving very fast through this pore. And the speed is around 10 microseconds per base. And the reason for this fast translocation is that the DNA strands are electrophoreted, pulled through this pore because there is a potential. This potential drop is required in order to measure ionic current. That's the sensing principle. But the consequence of that potential drop is that the negatively charged DNA is being pulled through the pore. So one has to use molecular brakes in order to slow down the movement of the DNA, and these molecular brakes can be enzymes, helicases, or DNA polymerases, and similar ones. And they dramatically slow down the movement of the DNA because they can be lodged to the top of, let's say, this protein pore, and thereby step by step, nucleotide by nucleotide, thread the strand through the pore. And one example of a readout is illustrated here. This readout was generated using an MSPA pore and the DNA polymerase. I do not show the pore here for reasons of visual consistency because I've shown previously the alpha-hemolysin pore, but the MSPA pore looks different. So this is the readout trace you get when you've got a trinucleotide repeat sequence TAC, 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 and you can see here in this more schematic and readout which was obtained from the raw data, that you can always get three steps corresponding to the three nucleotides. However, when there is a single nucleotide mismatch or mutation placed in here, so it's a G instead of a T, then one sees a couple of interesting things. First of all, we see that the current is changed for this particular base. It's now at this level, which hasn't been occupied before. But in addition, one also sees that there's a current deviation in the preceding and in the two following nucleotides. So in other words, the current blockade is measuring not only a single nucleotide, but also, well, let's put it this way, a single change in a base doesn't lead to a change in one current level position, but also influencing 2, 3, or even 4 or 5 other positions. So that's important because one can then get read accuracy from those single current traces. So one company, Oxford, so the companies are working on that, and one of those companies has been working on the commercializing the sensing principle is Oxford Nanopore technology, and they came out with a device called Nanion, and this product is basically composed of arrays of these channels, and these channels are then recording DNA sequence in high throughput fashion. This is the protein pore, which is used in the sensing devices from Oxford Nanopore. It's the CsgG protein pore, it's the bacterial pore, and my contribution was that I contributed to the characterization of this protein pore with electrophysiological techniques. So in this case, the Orbit 16 has been used. Now, the readout traces from the mini-orbit are more complex than in the previous slide where I showed a trinucleotide repeat. The real traces, they, when you really put DNA strands through, are highly complex, and one sees multiple different current levels, and that's due to the fact that the current level is influenced not by just one base position, but also by other base positions preceding or following that. Nevertheless, with machine learning algorithm, it's possible to infer the correct sequence out of those rather complex readout traces. And the application for portable sensing is has been published here, where this device has been used to read in real time sequences of Ebola viruses. Okay, so I've mentioned so far the sensing of DNA strands, the sequencing of DNA strands. Now let's move to the other analyte class proteins. You see on the right side the protein IgG, immunoglobulin, which is important in raising an immune response of humans and also other animals against foreign epitopes or invaders. The problem is that proteins do not fit inside the existing or protein pores used for DNA sequencing. So for instance, the alpha-hemolysin protein has a lumen entrance here of around 2 nanometers, and the other protein pores of around 3 nanometers, something like that. However, this protein analyte IgG has a size of 10 nanometers. There are a couple of tricks to sense proteins with small pores, and one trick is shown here, whereby a receptor is placed outside the lumen of the protein pore, and then the protein analyte can bind. There are other approaches to detect proteins outside a pore, but nevertheless, for a couple of applications, it would be highly desirable to have a protein analyte binding inside a pore. So it's better to get larger pores. Based on the properties of the polypeptide chains, it's rather difficult to de novo engineer large protein pores, large being with a diameter of 10 nanometers. An alternative route is to use DNA to build large pores, and one structure is illustrated here. It's a schematic drawing whereby those bluely shaded cylinders are representing double-stranded DNA, and those gray lines here at the top, these are DNA strands which are holding the DNA duplexes together. That's a schematic drawing. And as you can see here, it's a much bigger structure because DNA is basically a bigger bar polymer. And the diameter can be between 8 and 20 nanometers. Now, it's highly unusual to use DNA because DNA is the carrier of genetic information, but DNA is also a powerful material for rationally designed nanostructures. And this approach has been pioneered by Nat Seaman, whereby DNA oligonucleotides of specific sequence are chosen in such a way that then they fold, first of all, base pair, and then later fold into completely new structures. So in this case, the DNA oligonucleotides are assembled into a DNA cube, something which is not found in nature. And then Rothemund introduced the DNA origami approach, whereby a long black DNA oligonucleotide, a DNA strand, a scaffold, is then folded into complex structures with colored, short, staple strands. These are the colored ones. And this DNA nanotechnology is widely used, and it was exploited to build DNA nanostructures such as that. The bottom part here in purple, that's something which helps to bring the DNA structures to the lipid bilayer and make the DNA pores insert inside this bilayer. So these are two people in my laboratory who have been working on that. The construction principle for those DNA pores is illustrated at a very simple pore. This is a screenshot of the DNA design software. You can see that there are different panes, and in this right pane, one can say, well, I would like to have a structure composed of 6 DNA duplexes. These duplexes are represented as cylinders. And then I would like to have, let's say, a length of 16, height of 60 nanometers. And then this program automatically then calculates the sequences of the component DNA oligonucleotides. So we've got, if we look at the right-hand side here, differently colored parts in the cylinders. So these are the 2 strands within a DNA duplex, blue and yellow, but it can also be green and red. Now we switch to the left pane. On the left pane, we see that there are 6 numbers, which corresponds to the 6 duplexes, the 6 cylinders of the DNA nanopore. And each of these DNA duplexes is composed, first of all, of a blue strand, but then there's also a second complementary strand. Let's start with this yellow one here. And the yellow one is base pairing to this part of the DNA oligonucleotide, and then it's switching in a horizontal line to the next DNA duplex, number 2, where it hybridizes to another of those blue strands, and then later it switches to the 3rd DNA duplex. So these non-blue color DNA strands are providing the link between the DNA duplexes. Without those non-blue color DNA strands, the staple strands, the whole DNA structure would fall apart, but that's basically the glue between the DNA duplexes. Building such a DNA nanostructure is relatively easy, given that the design software is available, and also because the DNA oligonucleotides can be easily purchased from DNA synthesis companies. However, there's a problem, and the problem is that the DNA structure contains DNA, and the DNA is negatively charged. There's the phospholized backbone here, and here, and here. And the negative charged DNA structure is not going inside the lipid bilayer membrane or any other semi-fluid membrane, because the core of that is hydrophobic, so water and oil do not mix. So one has to use a trick in order to insert the DNA nanostructures inside the lipid bilayer. And one is to mimic nature and the natural biological protein pores, because they have got on their outside always hydrophobic amino acids. These hydrophobic amino acids on the outside of protein pores create a hydrophobic belt. And the idea was whether it's possible to mimic the idea of the hydrophobic belt and form a hydrophobic belt on the DNA, and that's illustrated here. The approach was to replace the phosphate, which is a phosphorus with 4 oxygens, with a phosphate analog, where one of those oxygens is replaced with a sulfur. This is a phosphorothioate. And given the unique chemical reactivity of the sulfur, the idea was to chemically modify the sulfur with a small molecule, ethyl iodide, which is then replacing the negative charge with a small hydrophobic tag. So a negative charge has been replaced with something charge neutral, hydrophobic. And in order to test whether this is actually possible, the DNA oligonucleotides were being purchased, carrying those PPT, the phosphothioate modifications at the right position. Then the DNA strands were chemically modified with the ethyl iodide, and then an equimolar mixture of those DNA strands was heated up, cooled down, and based on the self-assembly of those DNA strands, a structure was formed, and the structure could be visualized using atomic force microscopy. However, in order to prove that the DNA pore actually inserts inside a lipid bilayer, single-channel current recordings were conducted, and that's illustrated here. When a potential is applied, so when a positive charge is here, then ions are flowing to generate a positive current. And that's the schematic drawing. This was, I think, also the orbit 16 has been used. And here's Dr. John Burns, the person who has carried out this experiment in my laboratory. There are, of course, other types of lipid anchors in order to insert the DNA nanostructures inside. So this was the one I've previously shown, this hydrophobic belt. Other routes are to increase the size of the hydrophobic tags and thereby increase the hydrophobic pulling power, such as with porphyrin tags. In this case, only 2 porphyrins were required to get that DNA pore inside the lipid bilayer. And another type of lipid anchor is shown here, cholesterol, and Fitzsimmons' work, whereby the cholesterol are not here at the side, outside of the membrane spanning reach, but rather the cholesterol lipid anchors are attached to another part of the pore and are inserted from the top. My laboratory has spent a lot of time and effort in collaboration with colleagues, collaborators at other universities to understand the DNA nanopores in their interaction with the lipid bilayers. So, for instance, with Jakob Pieler, there was examined how those DNA nanostructures insert to the lipid bilayer. So there was it was found that there's a 2-step process. So in the first step, the DNA nanopores are just lying on the membrane, are adhering, and then in a second, slower kinetic step, those DNA nanopores are reinserting themselves to puncture the lipid bilayer. Then there's also a molecular dynamics simulation, which shows that for this particular DNA nanopore, so for the phosphorothioate modified pore, the lipid molecules in the lipid bilayer are rather close to the chemically modified DNA strands. So in this case, the DNA nanostructure doesn't lead to a major rearrangement of the thickness of the lipid bilayer, and there's a relatively tight seal between the DNA nanostructure and the lipid bilayer. However, some other DNA nanostructures and simulations from Alexei Aximentiev and Ulrich Kaiser, for instance, they've shown that in cholesterol-modified pores, this is not the case. The DNA nanostructures, the pores are not as rigid as protein pores. They are rather flexible, and that's illustrated in this simulation whereby the yellow lines represent the DNA strands in the duplexes, and with a bit of imagination, one can see in this structure here this 6-duplex bundle pore. The blue arrows here at the top and at the bottom, these indicate the movement of the DNA strands in those simulations. As one can see here at the top, the DNA strands are moving quite a lot, and at the bottom they are as well, so indicating that this DNA nanostructure is rather flexible. And that's also shown here in this IV plot, in the IV trace, where we've got recording the current versus voltage, and at relatively moderate potentials, plus minus 50 millivolt, we've got this red conductance state that's ohmic. But if the voltage is switched to 60 or minus 80 millivolt, then the pore switches to a lower conductance state, which is blue. And some of the dynamics illustrated in the simulations can be used to explain this switch. So I've shown you that the DNA nanostructures can be designed, that they can be formed, that they carry lipid anchors. I've also told you that the biophysical characterization of those nanostructures is important. I've mentioned two aspects, but there's a lot of other work gone into the characterization of these completely new structures. Now, we wanted to test whether the DNA nanostructures can be used to do something which would be very difficult to do with existing building blocks. So the starting point was that, well, those protein pores there are a little bit too small for protein sensing, so let's build the larger DNA nanostructures. And the beauty of those DNA nanostructures is that they are very easy to engineer given the simple base pairing, and also given the software which is available to design the structures. And we wanted to probe whether it's possible to also exploit DNA nanostructures for something else, namely to create a synthetic version of the biological ion channel. The biological ion channels, they carry out very important functions. The biological ion channels are involved in propagating the nerve signals. So, for instance, this is a ligand-gated ion channel at the synapse, where the neurotransmitter binds to this ion channel to a specific site. As a second function, the binding of this neurotransmitter is leading to a channel opening. It's illustrated here. And as a third function, this ion channel is able then to selectively transport some ions through the open channel. So these three functions, they are key for the biological ligand-gated ion channel. And for us, it was a template to test how good this DNA nanotechnology approaches, because for it's extremely difficult to rationally design from scratch protein-based ligand-gated ion channel, because based on the properties of polypeptides, the way they are folding, it's very difficult to predict and design artificial ligand-gated ion channels. And we said, well, is it possible to do that with DNA, given that it's such a wonderful building material? And this is our version of a synthetic ligand-gated ion channel, and the principle is illustrated here. We've got in blue the DNA duplexes, which are held together. So it's again a 6-duplex bundle DNA nanopore. In orange, to the left and right, are lipid anchors. These are cholesterol anchors, which hold the structure inside the gray lipid bilayer. At the top is a red lock strand, and this lock strand is hybridized to extensions in blue. These are DNA extensions, and they make sure that this lock strand is placed right at the top, and thereby blocking the entrance of the pore. However, this lock was designed in such a way that it is possible to remove this lock strand with a blue key strand via competitive hybridization. So this red lock and green key strand can form a DNA duplex, and by doing so, the red lock strand is being removed, thereby opening up the pore indicated by these arrows, which now go from the trans to the cis side of this membrane. The two states, the closed state and the open state at the bottom, they were characterized with with gel electrophoresis, so one can separate those two types of pores. They run at different heights in this gel electrophoretic analysis technique. And when one is measuring the current flowing through those pores, one can see that the open channel has a conductance of around 1.4 nanosiemens, and the closed one is around 1.6. So the closed one is not completely tight because there are tiny gaps between the DNA strands. For instance, here there's something white, but also the DNA strands itself. They are slightly permeable, at least here, for some small ions. Very beautiful single-channel current analysis is illustrated here. You see again the closed pore at the top and the open pore at the bottom, and you can see that in this IV ramp trace there is almost no variation in the channel. So previously in the IV ramp we had some lower conductance state, but this one is ohmic at -100 to +100, so it's very stable because this is a very compact structure. However, at the bottom you see the IV ramp trace for the open channel, and as you can see, the channel is behaving completely differently. First of all, there's more noise at negative potentials, but at positive potentials there's even more noise, strong fluctuation. And the way to interpret those fluctuations is to say, well, these DNA flaps, which used to be held static by this red lock, are now completely free, they are flexible, so there's no steric constraint holding those DNA flaps together. And when there's a positive potential at the other side, at the bottom of this pore, then electrophoretic forces move those negatively charged DNA inside towards the lumen of the pore, leading to the short blockade. But then due to the steric block, those two DNA flaps are flickering back, and then they're in this normal position corresponding to the top current, and then it goes on. So this is the characterization using the Orbit 16 device, and here we actually use the synthetic ligand-gated ion channel to selectively transport molecular cargo across membrane. And the assay is schematically shown here. We've got a vesicle, a lipid bilayer droplet, which contains some probe molecules, and the DNA nanopore is inserted, and the expectation is that if the key is added, then the channel opens, and one of those two probe molecules, only the green ones, will flow outside, whereas the other ones would stay inside. And based on the outflow of green but retention of red, one can then claim that this is selective transport. The probe molecules are illustrated here. These are the chemical structures. These are fluorophores. So that's a carboxyfluoroscene, and in green is a sulfurrhodomine B. The structure of carboxyfluoroscene and sulfurrhodomine B is very similar. The difference is that there's one positive charge here, whereas the carboxyfluoroscene has a negative charge. The bottom part of this fluorophore is, in terms of charge, the same. And the expectation or the hope was that based on the positive charge, this green sulfur B would go through because the channel in the lumen is negative charges from the phosphodiester backbone, and that the positive charge on this fluorophore would facilitate the transport, whereas in the red case, the carboxyferrocene, the electrostatic repulsion between the all negatively charged fluorophore and the DNA selectivity field would block passage. This is actually what happened. When the key was added to the pore, we saw an opening of the pore as confirmed by the flux of the green fluorophore. When there was a mismatched key added, so when this key was not perfectly matched to the red log strand, nothing was happening. And also here, when we look at the red fluorophore, so that's the negatively charged probe, there was no efflux at all. So confirming that at least for those small molecules, the DNA nanopore is charge selective. So I've indicated that the DNA nanopores can be used for many applications, including protein sensing. They can also be used for creating synthetic mimics of biological structures, such as the ligand-gated ion channel. I mentioned that DNA nanotechnology is also a very useful tool to create synthetic versions of protein cytoskeleton components, and there are a couple of very nice papers describing how the nanostructures can shape dynamically the lipid bilayer. Another application of DNA nanopores is to use them as cytotoxic agents or for drug delivery. I didn't go into detail. Okay, so the summary is that nanopore sensing is a very simple approach, requires only a tiny hole. It is possible to form handheld sensing devices, and the approach can sense and sequence DNA, detect proteins, and what was not mentioned can also be used to detect small molecules. I'd like to acknowledge the work, of course, of my group. This is a recent picture, so the people who have contributed to the work were Dr. John Burns, Genevieve Pugh, and Yongjin Sin. These are my collaborators, and I acknowledge support from these funding sources. Okay, many thanks for tuning in.